understanding of the renal Fanconi syndrome in cystinosis. Firstly, I would like to thank all the speakers before me. They have given a very great, elaborate discussion about cystinosis and the renal Fanconi syndrome. So I would just give a brief introduction about it. So what is renal Fanconi syndrome? It is a pathological condition of the proximal renal tubules of the kidney, in which solutes like glucose, amino acids, uric acids, phosphates, bicarbonates, which are generally reabsorbed in the blood, are not. They are actually excreted out in the urine. And it leads to several clinical manifestations like polyuria, excessive urination, dehydration, then feeling very thirsty. Now, what is the connection between renal Fanconi syndrome and cystinosis? Now, renal Fanconi syndrome can be inherited or acquired. Now, the most common form of this disease in the inherited case is because of cystinosis. And it is the first sign of cystinosis. Now, what is cystinosis? We all know it is a lysosomal storage disorder, which results in cystine accumulation in the lysosome. But what is the molecular link behind intrasomal cystine accumulation and the Fanconi syndrome? And children who have the Fanconi syndrome and cystinosis have a lot of problem in development, and eventually they suffer from kidney failure, and they have to go for kidney transplantation. So how did we come up with this particular study? Now, in 2005, a group called Miller et al., they did a high throughput screening where they identified different interacting proteins in yeast. And they found out the yeast form of cystinosis interacting with an exchanger protein called NHX1. And this study was then again done by our collaborator, Dr. Jean-Claude Khare, and he found the same results. So this basically led the foundation of our current study. So the questions which we have in our mind, which we plan to or hope to answer in the future is, the cystinosis is expressed in all the cells of our body. So why is it that the kidney gets affected first? Why is it that the renal Fanconi syndrome is the first manifestation of cystinosis? And all the cystinotic patients take histamine drugs, but that is not the cure for the renal Fanconi syndrome. So the mechanism behind it is not known. Now, as I've already stated, and we all know by this far, like what is happening in a Fanconi syndrome? Some solutes are not getting reabsorbed by our body. So probably there are involvement of exchanger proteins which play a role in solute exchange mechanism. So this study was so interesting because it actually found out an interacting protein, a transporter protein, which was interacting with cystinosis. And now we wanted to study the human form of this exchanger protein, which henceforth are 3. So what is NHE3? Just in brief. Now, there are 10 different forms of exchanger proteins in our body, in the human cells, but NHE3 is the most important one, and henceforth we have studied that more in details because it is more expressed in the proximal tubule cells of the kidney. And on the side, you can see a schematic diagram of the different exchanger proteins, but our study is concerned currently mostly with NHE3. And what is the role of the cystinosin protein other than just acting as a cystine transporter and its involvement in the renal Fanconi syndrome? So the study that was done by our collaborator, he found that the yeast form of the cystinosis is interacting with the yeast form of the NHE3 protein. And he did that by using an assay called the BIFC assay in yeast. So if I state this assay in just briefly, it's a very interesting assay. As you can see green colors all over. So what it does is, if you have 2 proteins, say A and B, and if you want to find out whether they are interacting or not, so what you do is you tag them with another protein, say X, which will give you a color green under the microscope. So you break that protein X, which will give you a green color under the microscope, and you tag it with these 2 proteins, and you put them in a cell. And if you get a green color, you're happy because you know that it's actually interacting. And that is what we saw in our experiments, that when we put a part of the green color protein with NHX1 and the other part with with ERS1, which is the yeast cystinosis protein, we found a green color in the dots, just stating that these 2 proteins interact. Now we wanted to expand our study in the mammalian cells. If it is happening in yeast, what is happening in the mammalian cells? So for that, we did an immunoprecipitation assay. So it is an assay where you can fish out 2 proteins or 3 proteins from the urine, proteins interacting with each other amongst millions of proteins. So to state it in simple words, you give some bait to the cell. Like, say, for example, if you want to find out if like my mom wants to call me and I'm not picking up my call, so the best bait for her will be to call my friend. And I can talk to my friend, right? So it's like that. So you basically design some molecules, you bait for one protein, like you try to target one protein, and if the other protein are very close and they play a role with each other, they will come along. So that's what we found. So in the scientific point of view, what we did was we used a tag, a green color tag, and we tagged it to NHE3 protein. And we used a red color to tag it to two different forms of cystinosis. It's just a normal and a long form of cystinosis. So we have the NHE3, which is green in color. We have cystinosis, which is red in color. And then we put them together in a cell. So we have 5 different type of cells. Now only the cells which have both the proteins will give you the band, basically in the scientific term. So only when both the proteins are together you get a signal, and you won't get a signal when only one of these proteins are there. This confirms that the proteins are interacting with each other. Now, to further confirm this, because we are scientists, we have to confirm each step we are going forward. So there was another assay called immunocytochemical assay, where we want to see whether these two proteins are localized. So if, as I said, if we tag one of the protein with green color and the other with red color, and if they are together, and we would expect a color like orangish yellow. And then when we saw that color under the microscope, we were very happy that actually these proteins are interacting and localizing in the mammalian cells. So now, what is happening in the patient samples, like in the actual world scenario? So we we isolated the proximal tubular cells from the normal patients, from normal healthy humans, and from the patient samples. And we stained the NHE3 protein with a green color. And you can see that in the normal cells, the color of green is in different in the normal cells, the NHE3 protein is expressed in the plasma membrane. So the pattern of green color expression here is very different from that in the cystinotic patients, because here it is mostly in the cytoplasm. So there's a difference in the expression pattern of the protein in the presence and in the absence of cystinosis, thus stating that cystinosis plays a role in the protein's function and in its localization. So from here, I can summarize the work that we are currently doing and what we have got. We've got that NHE3 interacts with the CTNS, both the forms of CTNS proteins. But now we are trying to figure out whether there are other molecules involved in the interactome. And also, we want to find out whether it is only this one particular exchanger protein which is involved in the interaction, or there are other exchanger proteins as well. So this would give us novel targets that we can address to in the future, design drugs against them, and give it along with histamine, so that even after histamine treatment, we have Renal Fanconi syndrome, we can treat that. So with that, I would like to conclude my work and thank our collaborator, Dr. Sergio Katz, who's spoken just some time back, and Dr. John Claude Khare. He is doing the wonderful work with the E-system. I would like to thank Dr. Stephanie Shirky and her lab for working there, and it is a beautiful picture of our workplace. And all my lab mates with all their help and support. Of course, the Cystinotic Research Foundation for giving us the grant and the money to work and do the cool stuff in the scientific world, so that it can be translated, and for bringing me as a part of the group. Sorry, actually, I have been working in cancer, but when I became a part of this group today, it's been an honor, and hopefully we'll find some answers to the problems. Thank you.
Thank you, Dr. Khare. And yes, welcome to our cystinosis family. The more, the merrier. We appreciate you. Does anyone have any questions before we take a break?
I was just wondering if you could explain a little bit more about what the NHE3 does in the normal human, in someone without cystinosis.
So normally, So NHE3 is a transport exchanger protein, and it helps in exchanging sodium potassium in the cells. So normally, in a normal patient, the sodium, the solutes, the glucose will be inside the cell. It will be, a lot of it will be reabsorbed and not secreted. Cystinosis happens, we see the Fanconi syndrome occurring, where most of the solutes are getting out in the urine. So from there, we had this thought that probably there's an involvement of cystinosis beyond just acting as a cystin transporter. And it might be one of these exchanger proteins. So, and we are yet to find out the details of it.
Do